The movement of microbes into hosts, infection, Chapter 14. The learning objectives for this topic are: describe the relationship between contamination and infection, identify and describe the portals through which pathogens invade the body, list the types of adhesion factors and the roles they play in infection, explain how a biofilm may facilitate contamination and infection. The overview of the topic is exposure, contamination and infections, portals of entries, and role of adhesion in infection. First off, let's talk about the difference between contamination and infections. And I'm going to give you the book's definition first, and then I'm going to tell you a little story that will help you remember the difference. So, contamination is the mere presence of an organism. Infection is successful invasion and colonization of a human body. So, here's my little story. I have a cold. I have adenovirus. And I'm sneezing my brains out. And I sneeze into my hand right before I shake your hand. Well, you are now contaminated with adenovirus. Does that mean that you are infected with adenovirus? No. Adenovirus is not set up to move through skin to infect you. So if you go and wash your hands thoroughly, you get rid of the contamination and you don't become infected with the adenovirus. Now, right after you shake my hand, if you rub your eyes, then you can become infected, because adenovirus is set up to grab a hold of the receptors on mucosal cells, like what are around your eyes and in your nose and your mouth, and they can get in and infect cells and give you a cold. So that's the difference between contamination and infections. Now, how the contaminants get into you to potentially cause infection. This figure here shows all of the different portals of entry for humans. One thing to keep in mind in organizing them is that most of them are mucosa. You've got the conjunctiva, you've got the nose, you've got the mouth, you've got the urethra, you've got the anus. Those are all mucosal. Now, you can have portals of entry if you get broken skin or if an insect breaks your skin. And then for the babies, the placenta can be a portal of entry from the mother to the fetus. Another thing to keep in mind is that pathogens have a preferred portal of entry. Now, in my example of the adenovirus, it prefers the conjunctiva of the eye. If it can't get that, it'll go with the nose or the mouth. But it really doesn't want broken skin or an insect bite. It's just not happy doing that. It's also not able to cross the placenta. It has a preferred portal of entry. It wants to get into the respiratory tract because it causes a respiratory disease. Now, for gastrointestinal pathogens, they prefer to enter through the mouth, go through the stomach, into the intestines, and cause gastrointestinal distress. Sexually transmitted organisms prefer to enter through the vagina or through the penis. It makes sense. Once the pathogen has gained entry into the body, it has to be able to grab a hold. Now, remember with viruses, we talked about receptors and how the viral receptors match up with the cellular receptors on the host cell, and that's why most viruses have a very narrow host range. Well, guess what? Bacteria have receptors too. So do protozoa. They don't have hands. They can't actually grab a hold, so they have to use molecules that will grab a hold of receptors on the host cell. In bacteria, we call this a ligand. The receptor on the human host is usually a glycoprotein. So think of it the same way as how viruses do it, when they're adhering to the cell. Now, biofilms are groups of cells that have established themselves someplace. In nature, it can be on a grain of sand, on the surface of a rock, you name it. In humans, this can be on the surface of your respiratory ciliated cells. It can be on the surface of your intestinal cells. They build up these biofilms, produce kind of a slime layer on the surface, and that protects them from your immune system. It all works together. It also can protect the pathogen from antibiotics. Antibiotics have a hard time getting through the sticky outside layer to where the actual pathogen is. That's it for this topic. Here are reminders of what the learning objectives for this topic are.